right, hi. So my name is Cécile FORMOSA DAGUE. I'm a researcher in the Toulouse Biotechnology Institute in Toulouse, obviously. And today I'm, sorry. So I have to stay here though, because they put me this, but it doesn't work. Okay, because usually I move it, okay. So, oh yeah, sure. Thanks. So today I'm really happy to present you the work that we have been doing with my colleague Maria Areco, who's from the University of San Martin in Buenos Aires in Argentina. And this work is about understanding how microalgae can actually remediate heavy metals using a biophysical approach. So I'm not going to talk too much about that because previous speaker already told you about the Reconquista River in Buenos Aires. And so I have the map, actually. So you can actually see that the river goes through this very dense populated area of Buenos Aires, and it's a heavily polluted river. You can see the pictures, but it's also containing a lot of heavy metals, high level of heavy metals, and that's obviously a problem. So what the team in Argentina did is that they took samples of this river, and they found out that actually microalgae cells could grow in the water of this river even if it contains high levels of heavy metals like zinc. And they isolated one species in particular that is called Parachlorella kessleri, and which is able to withstand those high levels of zinc. And then they tried out different conditions, and what they found is that when you grow the cells with different nitrogen source, they will be able or not to absorb and remediate the zinc from the culture medium. And so you see when the cells grow in the presence of ammonium, the zinc concentration in the medium stays the same, but if you grow the cells in using nitrates, then the zinc concentration is going to decrease because the cells are able to remediate the heavy metal. And so the question was, how can they do that? So to answer this question, we used atomic force microscopy. So perhaps I'm just going to say a few words about it because maybe you're not familiar with it. And so it's a type of microscopy that allows you to probe living cells but also to probe their interactions with their environment. And so basically you have here a piezo that can move in the Z direction with a very high precision of the angstrom. And mounted on this piezo you have a cantilever. At the edge of it you have a tip. And basically what the tip is going to do is that it's going to approach, touch the sample, indent in it, and then be retracted. And all the time you can follow the movement of the tip using a laser system that is reflected here on the cantilever and then on the photodiode. So this mode of operation of atomic force microscopy is called force spectroscopy. And so basically it gives you access to force curves that you can get a lot of information from. And so you have here the beginning of the force curve, so the tip is away from the sample. It will go down, and here it touches the sample, and then it will continue to push in the sample, to indent in the sample, and from this part of the curve then you can see the resistance of the sample to the compression from the tip, and then you can deduce the rigidity of the sample, if it's soft or if it's rigid. And then when the tip is retracted from the sample, maybe there will be an interaction between the tip and the sample. And because you keep on pulling on the tip, at some point this interaction will break, and this is visible on the force curve here by those peaks that we call adhesion peaks. Okay, so that's what you need to know to understand the rest of the talk. So there are some advantages of using this technique. For example, it operates in liquid. That's a big advantage because we can work on living cells. And also, it's a high-resolution imaging technology, but better than electron microscopy because, we're live, right? And then because we can do those force measurements, we are able to access both the mechanical and the adhesive properties of cells. All right, so let's go back to the cells, Parachlorella. So the first idea was to find the difference between the cells that were grown with nitrates or with ammonium. And so what they did is that they did some staining, and here you can see that in fact the cells that have been grown with nitrates are surrounded by a thick layer of EPS. I never remember what it means. Extracellular. All right. Thank you. You got it. And when the cells have been grown with ammonium, you can see that we don't see these EPS around the cells. So this was confirmed by AFM imaging. I'm moving for the shadow. So you can see, maybe it's not so clear with the lighting, but you have the cell here, and basically you have here a layer of material that surrounds the cells that we cannot see in the case of NH4 cells. And also the edges of the cell here is super blurry, and we don't have a good resolution because, in fact, the EPS, they cover the surface. Okay, so that's observation, but to be really, really sure, we then did mechanical analysis. And so basically, this is what I told you. We approach the tip from the surface and we keep pushing in the cell. And so when we do that with NO3 cells, you can see that we have a super long indentation, which means that there is a big deformation of the surface as we push in it. But when we do that with the NH4 cells, I mean, the force curve is almost vertical, which means that there is basically no deformation of the surface. And so this can be explained really easily, because then the EPS usually form a very soft gel-like structure, and that's why they're super soft. And so this confirms that NO3 cells are indeed covered by this big, thick EPS layer. And in the case of NH4 cells, we actually directly touch the cell wall, which is a rigid structure. Okay, so we also quantified this using Young modulus. So Young modulus is the parameter that will illustrate the rigidity of the cell wall, of the surface. And with NO3 cells, we have approximately 500 Pascal, so very, very soft. And then with NH4 cells, we increase, you see there's a break here, to 800,000 Pascal. So, I mean, it doesn't even compare. All right, so now we know that the difference between NH4 cells and NO3 cells is that NO3 cells produce EPS and NH4 cells don't, okay? So how are these EPS involved in the remediation of the zinc? So to answer this question, we repeated the 4 spectroscopy experiments, but in this case, what we did is that we directly put the zinc particles on the tip using a protocol that we developed in my team some time ago. And so basically we just glue the particles on the tip, and we can obtain like that the zinc modified AFM tips. And you can see here the same image. This is the cantilever, this is the tip, and here when we have put the zinc particle at the edge of it. And then we use these zinc modified AFM tips to directly probe the interactions with the different types of cells, so NO3 or NH4, okay? So when we do these experiments, these are the results that we get. So basically with the NO3 cells, if you remember the first slide on AFM, we have force curves that present a lot of retract adhesions. So basically what it means is that there's a lot of interaction between the cell surface and the tip. And also what is interesting, I find, is that these unfoldings, they take place over very long distances, so 20 micrometers. That's the size of the force curve here. So this is, I mean, it's amazing, right? To unfold something that is 20 micrometer long from a cell that is actually 10 micrometer in diameter. So this was actually super fun experiments to do for this. And then when we repeat that with NH4 cells, we can see that there is no more retract adhesions, and the few retract adhesions that we see, they're very small, which means that the interaction force between the tip and the sample is pretty low. And so basically what it means is that the zinc that is on the tip is able to interact with the EPS at the surface of the NO3 cells, but not with the cell wall that we directly touch when we, yes, okay, when we have the NH4 cells. We can also quantify this, and so what we do to quantify is that we measure here the work of adhesion, which is the area under the curve, and which represents the quantity of energy that you need to actually detach the tip from the sample. And so you can see that within the 3 cells, we have a very high work of adhesion that is almost equals to 0 within each 4 cells. And so this is like the ultimate proof that zinc is able to interact with the EPS but not with the cell wall. And so that's how the cells can remediate the zinc. It's because their EPS can absorb it from the culture media. So to conclude, we isolated Parachlorella from the Reconquista River and show that these cells could remediate zinc only when they're grown in the presence of nitrates. And in these conditions, they actually produce EPS. Those EPS can interact with the zinc and absorb it, and this is how it works. And so basically what it shows here is that we can use a tool like atomic force microscopy to actually understand the mechanism of the biosorption that we observed. And also, I mean, of course, it shows the potential of Parachlorella to bioremediate heavy metals. And with that, I thank you for your attention.